Hello everyone, welcome back to the channel. In today's video, we are going to understand one of the most important bioinformatics tools used in molecular biology, that is Nucleotide BLAST, also known as BLASTn. This tool is widely used to compare DNA or RNA sequences and identify their biological significance. So let us begin. Nucleotide BLAST, or BLASTn, is a bioinformatics tool developed by NCBI. It is used to compare a query nucleotide sequence, such as DNA or RNA, with a large database of known sequences. The main goal of BLASTn is to find regions of similarity, which helps us identify genes, organisms, and evolutionary relationships. The image presents a conceptual overview of Nucleotide BLAST, BLASTn, as a continuous analytical workflow, illustrating how raw nucleotide information is transformed into meaningful biological insight. Rather than emphasizing procedural steps, the diagram visually integrates the core components of BLASTn into a single, coherent narrative of sequence analysis. At the foundation of the image is the query nucleotide sequence, representing DNA or RNA obtained from experimental or sequencing efforts. This sequence is the primary biological input, typically derived from PCR products, cloned fragments, or high-throughput sequencing reads. Its placement at the beginning underscores that BLASTn is fundamentally driven by experimentally generated nucleotide data. The central portion of the image highlights the NCBI BLASTn engine and its interaction with major nucleotide repositories such as GenBank, RefSeq, and the non-redundant NT database. This emphasizes BLASTn's role as a comparative tool that searches vast, curated collections of nucleotide sequences to identify statistically significant similarities. Conceptually, this stage represents large-scale pattern matching, where local alignments are detected between the query and database sequences based on sequence similarity and scoring algorithms. The image then transitions to the alignment results, which are shown as tabular metrics and graphical alignment bars. These outputs convey the quantitative backbone of BLASTn interpretation, including percent identity, query coverage, and e-value. Together, these parameters define the strength, extent, and statistical reliability of each match. The graphical alignment view reinforces the idea that BLASTn does not merely return names of sequences, but provides a visual and numerical representation of how nucleotides correspond across sequences. Finally, the rightmost section of the image connects computational results to biological interpretation. Icons representing gene identification, species matching, mutation detection, and evolutionary or functional inference illustrate how BLASTn outputs are translated into real biological conclusions. This reinforces a key conceptual point. BLASTn is not an end in itself, but a bridge between raw sequence data and biological understanding, enabling researchers to annotate genes, confirm organismal origin, and detect sequence variation. Overall, the image effectively communicates BLASTn as an integrated bioinformatics framework, linking nucleotide sequences, curated databases, statistical alignment results, and downstream biological insights into a unified analytical process widely used in molecular biology and biotechnology research. It's the website. You just reach here by searching at Chrome NCBI, and you just look at it, and go for the nucleotide sequence, or you can go for gene. Here it is, the search results, different organism, different strain. We go for beta-galactosidase gene. You can go for gene, beta-galactosidase, in E. coli. You can get other information: location of the gene, information about operator, promoter region, enhancer region. For Nucleotide BLAST we need the FASTA sequence, or you can directly run BLAST from the website. Here it is, the FASTA of the gene sequence. You can copy and paste it or directly run BLAST. And if you scroll down you will get other information. As I click the Run BLAST option, so I jump into this page directly, you can see the accession number of the gene, and now we can go for BLAST. Just check once the default parameters of the BLAST. You need to wait for some time for the BLAST result, 2 minutes approximately, then here it is, the result. Now it is the result page, sequences producing significant alignments. At the top of the page, the heading, Sequences producing significant alignments, indicates that all listed entries have passed BLAST's statistical threshold for meaningful similarity. These are not random matches, they are biologically relevant alignments based on scoring matrices and probability calculations. Each row in the table represents one database sequence that aligns with the query. Description column. The description column provides a short annotation of the matched sequence. In this case, most entries are annotated as Escherichia coli strain, chromosome complete genome synthetic construct clone. This strongly suggests that the query sequence originates from or is identical to a region of the E. coli genome. The presence of many different E. coli strains indicates that the sequence is highly conserved across strains. Scientific name. The scientific name column confirms the taxonomic identity of each hit. Here, nearly all matches are labeled Escherichia coli, with one exception being a synthetic construct, which usually represents a cloned or engineered DNA sequence in a laboratory vector. This taxonomic consistency reinforces the conclusion that the query sequence belongs to E. coli rather than a related species. Max score and total score. The max score represents the highest alignment score for a single alignment segment between the query and the subject sequence. The total score is the sum of scores from all alignment segments. In this result set, the max score and total score are identical, which means the alignment is continuous and uninterrupted, not fragmented. This is typical of a full-length, high-quality match. Query coverage. The query coverage column indicates how much of the query sequence is included in the alignment. A value of 100% query coverage means that the entire query sequence aligns with the subject sequence. This is a strong indicator that the submitted sequence is complete and correctly identified. E-value, expect value. The E-value is one of the most critical statistical parameters in BLAST analysis. An E-value of 0.0 means that the probability of obtaining this alignment by chance is effectively zero. In practical terms, this confirms that the alignment is extremely significant and biologically reliable. Percent identity. The percent identity column shows the proportion of nucleotides that are exactly identical between the query and the matched sequence. Here, the value is 100%, indicating a perfect match. This means there are no mismatches, insertions, or deletions between the query and the aligned region of the database sequence. Accession length. The accession length refers to the total length of the subject sequence stored in the database. Since most hits are entire chromosomes or complete genomes, the accession lengths are very large, typically several million base pairs. This does not mean the query sequence is that long, rather, it aligns to a specific region within those large genomic sequences. Accession number. The accession column provides a unique identifier for each sequence record in the NCBI database, such as CP170318.1. These accession numbers allow researchers to retrieve the full genomic record, annotations, and metadata for further analysis. To conclude, Nucleotide BLAST is a powerful and essential bioinformatics tool that allows us to compare nucleotide sequences and extract meaningful biological information. Understanding BLASTn is very important for students and researchers working in genomics, molecular biology, and biotechnology. Thank you for watching this video. If you found this explanation helpful, please like, share, and subscribe to the channel. If you have any doubts or want a video on protein BLAST or BLAST result interpretation, let me know in the comments.